Okay, so today I'm just going to do a diagram for bacteria, and I'm going to use the shape and the gram staining for the bacteria to make this diagram. So with bacteria, just so you know, they usually come as different shapes. So it could be cocci, rods, it could be branched, it could be spiral shaped, different types. So that's the shape, and then the gram staining. It could be either positive staining, negative staining, or it could even be something called acid fast staining. So there's different types of shapes and different gram staining. So to begin, instead of looking at all of this and having other factors such as if it has spores or it doesn't have spores, like non-spores, we're gonna simplify the diagram initially by just looking at these four things. So what it's gonna be is you're gonna have gram positive, and it could either be cocci or it could be rods. And then we're gonna have gram negative, and then this could be either cocci or rods. And then with this, we can look into them more specifically to see the different types of species and everything. So to begin, I'm going to first do the diagram for the gram positive. So the gram positive, as I said, is going to be here, gram positive. And you can either have cocci or rods. So this is the two shapes that you could possibly have. Now within the cocci, you have two different types of cocci. You have staphylococcus or streptococcus. And how would you differentiate between the two? There's a reaction called the catalase reaction. So if it's catalase positive, then it's staphylococcus, and if it's catalase negative, it's streptococcus. And within the staphylococcus, you can have different species, and they can be categorized them to either coagulase positive or coagulase negative. So the positive will be over here, and the negative will be over here. So coagulase positive, you have Staph aureus, and for the negative you have Staph epidermidis and Staph saprophyticus. Okay. And then for the streptococcus, you can categorize them depending on hemolysis. So hemolysis is what happens when you put them in blood agar. So with this one, you have three categories. You're gonna have alpha. The thing is, with this category, it's good to use the color that you'll see on the blood agar. So you see like a greenish color for alpha. For beta, a yellowish color, so I'll use yellow here. And for the gamma, it's going to be unchanged, so I'm going to use red. And the bacteria for this is Strep pneumoniae, Strep viridans, Strep pyogenes, Strep agalactiae. Now, for the gamma, I'm going to put Strep bovis and also something called enterococci. But the thing is, I'm gonna underline enterococci. The reason for that is because, I'll put it in a, I'll put in a box, is because it could be alpha, beta, or gamma. It could be any of the three. So technically it's not gamma. And the reason why I put it here is because even though you can categorize them as hemolysis, you can also categorize them as groups. So for instance, strep pyogens is group A. So I'm just going to write A, B here. So this is A, this is B. So group A, streptococcus, are strep pyogens, and group B are strep agalactiae. And group D is strep bovis and enterococci. That's why I put these two together. Okay, so the reason why I'm drawing this diagram like this is because you like to have a big piece of paper, so then you can have this diagram here and then write individually what these bacteria can do. So for instance, on the diagram that I've made before, I put S. aureus here, and I've written some things about it. So for instance, leading cause of gastroenteritis, leading cause of infection during surgery, and for instance, osteomyelitis, it can cause osteomyelitis. But here, okay, so this is just some examples of some things that you can write for strep aureus, and I'm not going to go over all the things that you could write about all the bacteria because you can find them in the book. But things that I like to point out, for instance, is you'll see leading cause of gastroenteritis, and you should know it's about 2 to 6 hours. Why is that important? Because when you ingest other bacteria, it'll take a day or so for you to get like vomiting, etc. But with Staph aureus, it'd be very quick. It would be within the same day, 2 to 6 hours after. And the reason for that is because instead of ingesting the bacteria, you ingest the toxins, and that will give you a quicker response and you'll vomit sooner. And for instance, osteomyelitis, it can be caused by Staph aureus, or it can be caused by salmonella. But for salmonella, it's usually found in sickle cell patients. So not sickle cell here. So here, Staph aureus, you just write down certain things. So things that it causes, the toxins that it has, etc. And the drugs that it could be used to kill it. Like you can talk about MRSA, etc. and vancomycin, killing that one. Anyway, so Staph aureus is here. You then have Staph epidermidis and Staph saprophyticus. So here, if I just draw a diagram, you have your Staphylococcus like so, and you can write as epidermidis causes endocarditis, etc. This causes UTI, sexually active people. Now the thing is, you can see that within this diagram, you can work out from a case what the bacteria is. So if they say that it's gram-positive cocci, and it says, for instance, it's catalase positive, you know it's going to be Staph. Then it says it's coagulase positive, then you know it's going to be Staph aureus. But if it's catalase positive and then coagulase negative, it could be either Staph epidermidis or Staph saprophyticus. So how are you going to differentiate between these two? Well, there's gonna be, so I've written these two here, and there's these two. They're categorized into two, but you can use drugs to differentiate between the two. So I remember it as knob, N here, O here, and B, because that's the name or the first letter of the name of the drugs that can differentiate between the two. So for instance, the N stands for novobiacin. Novobiacin, so I'm just going to write that down here. So novobiacin. Epidermidis. This epidermidis is sensitive to it, so it will kill epidermidis. But if you use novobiacin for saprophyticus, for instance, it will be resistant. So novobiacin for saprophyticus is resistance. So as I said, how do you differentiate? Catalase positive, coagulase negative, and then it says that the novobiacin resistant, for instance. So that means you know it's saprophyticus. That's how you differentiate. And then let's give you another. So let me write out the other things here. So you're gonna have strep pneumo here. Put the box, and then you write your information here. Then you can have viridins here, and you can write information here. Viridins is a group name for bacteria, so if you see, for instance, strep mutans, same thing. So you know that causes endocarditis, usually in you find this in the mouth, so dental procedures can cause endocarditis. Okay, so I should have made this bigger, but so you write whatever here, and then you're gonna have another thing, and you're gonna write aspergins here, put it in a box, and you write information, and S-A-galactiae, put in the box, and then you write information here, and then you do the same over here, bovis and enterococci. Now, the drug for NOB, so N was novobiacin, for O is going to be optichin, so I want to use green because these are sensitive, so they'll be killed. These are resistant. So this is called O, so optochin, and that's meant to be red. So optochin, and this is called bacitracin. And same drugs over here, optochin. I'm totally using the wrong colors. Okay, so you get the drift though. Optochin here and bacitracin here. And same over here. Just gonna write speech marks. Okay. So again, another case study. You have catalase negative. You know, strep. You say hemolysis alpha, hemolysis, and then you use, for instance, optochin. If it's optochin sensitive and it gets killed, then you know it's pneumo and not viridins. Okay. So now we're going to talk about rods. Rods, on the other hand, you have Bacillus, Clostridium, and you're going to have Corynebacterium and Listeria. Now, these two are spores, so you can find spores for these, and these are non-spores. So gram-positive rods, you can have, let me put this in a box. You can have spores, so it can be Bacillus or Clostridium, and Corynebacterium, you can have, non-spores, you can have Corynebacterium and Listeria. And along with these, you have, you can have some bacteria which are not rods but actually branched. So branched ones are actinomyces and nocardia. Yeah, so these two will be branched. And then how do you differentiate between these two branched ones? Well, this one is something called acid fast, or it's partially acid fast actually. Nocardia is actually partial acid fast, and actinomyces is not acid fast. And I'm just going to add one more bacteria called Mycobacterium. And you guys know that one causes TB, and that's acid fast. Acid fast. Okay. So here you have the diagram at the top, and then underneath them you have description of all the bacteria. And for bacillus, for instance, you can write information over on the side. So bacillus, but you have different types. So bacillus anthrax, this bacillus cereus, for instance, and then clostridium. So this is on the side, you write your information over here for this one, information for this one. Clostridium, you can have, you know, this acid fast. So I'll try red here. Clostridium, you have 4 different types. The first one, you can write Clostridium tetani. Second one, Clostridium botulinum. Sorry if I spelled them wrong. I don't know if you can read my handwriting anyways. Clostridium perfringens. And fourth, you have C. difficile. Yeah. And Corynebacterium. I spelled it wrong. I'm just quickly writing it out. So just to let you guys see what the diagram should look like in the end. So Corynebacterium, diphtheria. Well, I didn't write diphtheria here. Yeah, just write diphtheria here. And then you write information here. Listeria, monocytogenes. And then write information here. And then you can just, I don't know, have a little box. This is where your branched bacteria are. So actinomyces, write information, nocardia, and Mycobacterium. So the reason why it's good to have a diagram, not just the diagram, but the information as well describing each of the bacteria, is that you can visualize the gram-positive bacteria as a whole, and you don't have to look at them individually and remember, oh, what do I know about strep Acalactia, for instance. You know that in a diagram it's here under the beta hemolytic streptococcus. Another thing is that strep bovis and enterococci, how do you differentiate between the two? You can grow the medium in 6.5 salt, and just like always, I put it that way wrong. But enterococci, so let me just switch now. So enterococci, coccus, can, so I'll put that in green, can grow, and S. bovis cannot, so that's red. No. And that's how you differentiate between these two. So again, gram-positive cocci, catalase negative, and it's a gamma hemolysis. Then you can use 95% salt, and if it is, then it's probably enterococcus. Rods, there's these 2 spores. These are non-spores, and these are branched. Two spores, 2 non-spores, 2 branched, and then Mycobacterium is acid fast. So that's how you would differentiate the bacteria if you had a case and it described the bacteria to you. And yeah, you can have, then you can fill out these things yourself. So for instance, I don't know, something that I remember, listeria, it likes the cold, so you can find this in the fridge, for instance. You can find keywords, so Corynebacterium looks like Chinese letters, and it gives you what is it, diphtheria throat. So you see like a black fibrous mass on the back of the throat. And then what happens to the throat? So you look at the throat, you have a little black thing on the throat, but even though the bacteria are found in the throat, it releases toxins to two areas. So it releases toxins to the brain and to the heart. So you know that affects the neurons in the heart. So this is just like an example of something that you should write down for this bacteria. But yeah, this is the gram-positive